Okay, I think the time is almost 7:15 now. So
By the way, do you have many Australians there?
studying, yeah, but in faculty of language or social culture. Yeah, we have. I think a lot of Australian would like to study Bahasa Indonesia, Prof.
Yeah, looks like a nice place.
Yeah, that's why we also have Australian here.
Good morning, Philip.
Good
Prof.
morning.
Irfan, this is Prof. Irfan.
Morning.
Yeah.
Good
So
morning.
Can
Thank you very much
I,
for
oh, you're most welcome.
thank you
Can
very
I?
much sparing your time for us.
Oh, my pleasure. My pleasure.
okay, maybe should we start now?
Can I just confirm that you're seeing the full screen again, not the
Yep.
presenter mode? Okay,
Yeah.
great.
You
Yep,
full
wonderful.
screen, Prof. Philip. Okay, so we have 96 attendance today, but I believe it will be getting more and more. But it's 7:15 now in Indonesia. Okay, let's open then. Okay, good morning, ladies and gentlemen. Hello, everyone. You are most welcome. If you would like to turn on your camera so Prof. Phil can see you guys. How beauty and how handsome you are, students.
Terrific.
So, thank you very much, Prof. Philip Hugenholtz, the director of Australian Center for Ecogenomics and also lecturer at the University of Queensland, Australia. It's really our honor today, Prof. I know that you are so busy, but you still have time for us, for Indonesian student lecturers and also staff, because today will be not only from our university, Universitas Gadjah Mada, however, some for other universities, and even you mentioned before Surabaya, and I bet that we have Surabaya. Oh, Prof. Irfan is from Surabaya, Profil. I just remember. Prof,
Nice.
yeah, Profil was asking that is there anyone from Surabaya this morning, Prof. Irfan?
Yeah,
Yeah.
I'm
So
from Surabaya.
from Surabaya, yeah. So before we start, Prof. Irfan, please welcome this guest lecturer. The time is yours. The screen is yours.
Thank you very much, Desi. Thank you very much, Philip, Professor Philip Hugenholtz. For us, evolution is now became a subject, compulsory subject for student from the Department of Agriculture and Microbiology. So inviting you and you accept our invitation to have this lecture, a very honor for me. And I hope that this is not the end or the last. Hope that this one is at the beginning and it will be continue for more and more lecturer coming from your side, even for you yourself. We are waiting for next collaboration, either in the academic as well as for research collaboration. Thank you very much. So, as I also have a lecture today, class today, so I hope that your lecture will be nice and also have a good result for the student. Thank you very much.
Thanks very much for that very kind introduction. Yes, I think that Australia and Indonesia should collaborate more scientifically, seeing as we are neighbors. So that's a good thing that we should be doing together. So I'm going to talk today about the interface between ecology and evolution. And so generally, you have people that are interested in the ecology, and like they might be interested in gut microbiome. So the two examples I'm going to give you today are to do with the rodent gut microbiome and the marsupial gut microbiome. This is what the AI came up with, the ChatGPT, but actually I'm going to be talking about koala, not kangaroos, but I like this picture the best. And And often you'll see people that talk about the ecology, microbial ecology, don't really think about the evolution, and vice versa. You'll have other people that are interested in evolution of, say, bacteria, but then they don't really think about the ecological setting. So the main message I wanted to get across to you today is that it's actually very powerful to look at both sides of the same coin, because one informs the other. So I'm going to tell 4 shortish stories, 10 or 15 minutes each, to sort of illustrate this. So the first story I'm going to talk about is a bacterial group which was only known by the name S247. So I think the students there are familiar with the concept that most microbial diversity hasn't actually been grown on agar plates. So much of it has been resistant to being cultivated. And so we've started to learn about this uncultivated diversity over the last 30 years or 40 years, beginning with using genes such as the 16S ribosomal RNA gene. And as the sequencing technology improved, now we can see them via their genome sequences. Even if we can't culture them, we can investigate their genetic blueprints. So this is one of these large conspicuous groups called, which was only known as S247, which was named after a 16S ribosomal RNA sequence. It's in the phylum Bacteroidota, which used to be called Bacteroidetes, and in the order Bacteroidales. And it's quite conspicuous because there's many representatives. I'll show you that in a minute, and no cultured representatives at the time. So this is the work of Kate Bauman, who's a postdoc here. She doesn't like cameras, so it's very hard to get a picture of her. So that's why she's just here as a silhouette. She avoids cameras a lot. And so what was very conspicuous in terms of the ecology of this group S247 is that they're found almost exclusively in the gastrointestinal tract or guts of warm-blooded animals. So in 2016, when we wrote a paper about this, we gave it the name Homeothermaceae to reflect the fact that it's found in warm-blooded animals. If you look at the 16S databases like Silva, there are 2,800 near full-length 16S sequences. If you look at the genome databases, and you mentioned that the students might be familiar with the GTDB, the genome taxonomy database that we maintain here, there are actually almost 25,000 genome sequences. So it's a hugely represented group. And these, according to the GTB taxonomy, represent 32 genera and 611 species. So a huge amount of diversity. If you look at their genomes, the genetic blueprints, it looks that they're primarily fermenters, and they can produce acetate, propionate, and succinate as the end products of the fermentation. So they're similar in that respect to other members of the Bacteroides, and are referred to use fumarate as a terminal electron acceptor, which is pretty common. They're also inferred to be nanorobes. So this is the new classification of aerobic metabolism. That means that they can survive in the presence of small amounts of oxygen, and in fact, probably use it. And probably most striking about the group is that they have a lot of enzymes for breaking down complex carbohydrates. And so based on this, Kate inferred that there were 3 potential trophic guilds or feeding guilds, based on the presence of these carbohydrate active enzymes. So they're the ones that break down starch using alpha glucanases. Then you have a group that look like they're geared towards breaking down plant glycans, and then a third group that are more specialized in breaking down animal host glycans. And so I didn't really talk about how to read this graph, but this is a heat map showing that these are plant. Can you see my mouse, by the way? Let's have a look.
Yeah, we can see.
Pointer options. So I'll go for laser pen. Laser pointer. There we go. It might be easier to see that. So on the horizontal row, they show the individual types of enzymes, carbohydrate active enzymes, GH. For these are common carbohydrate active enzymes, and these ones down here are more common in to breaking down plant material, and then these ones here are used to break down animal host glycans, and then this is the amylases here for breaking down starch. And as you can see, you find them in a wide range of different animals: baboon, cow, guinea pig, human, koala, mouse, rat, and sheep. If you look at those trophic gills on a tree of the group, you can see that you have clusters of some that are specializing in certain types of plants. Like this group here is breaking down plants. This group here are just breaking starch down. But you also see quite a bit of mixing between different groups. So some members of a particular genus, either different genera, specialize in breaking down plants. Others are breaking down animal glycans. Feel free to stop me if there's any questions along the way. I think that's okay, Bessie, that's all right?
Yes, you are, Prof.
Okay, great. I'm happy to answer questions as we go.
Yes. Sure.
So this S24/7 group is most conspicuous in mice. It's very, very high relative abundance in laboratory mice mainly. 10 to 50% of the sequences obtained from the lab mouse gut are actually belonging to this group. In humans, it's in urbanized humans anyway, it's usually around 2% of gut microbiome, but in some populations can be extremely high. So for instance, in Hasda hunter-gatherers, which we are very famous from the point of view of human gut microbiomes, they're up to 70% belong to this family, the S24/7. The first isolate from this family was actually obtained from the mouse gut from Thomas Clavell's group. It was short. That was actually done pretty much around the same time that we did our molecular analysis back in 2016. And they called the family Muribaculaceae, which is from Muribaculum because it's rod-shaped, and muri meaning it's coming from the mouse. And then intestinale was the species they described pertaining to the intestines. So it's actually a pleomorphic rod. So if you look at it under the microscope, you could be forgiven for thinking that it's a mixed culture, but it's actually a pure culture. This is a characteristic of this organism. So consequently, because there was an isolate and the way that the nomenclature works, the name Muribaculaceae has priority over our name, which I thought was very nice, Homeothermaceae. But now if you look at it in the literature, the group was formerly S24/7. It's called Muribaculaceae. Yeah.
I have a question before that. You mentioned that it is not this is pure culture.
This is a pure culture, yes. So this is the first pure culture that was obtained in 2016. Up until that point, there were no cultures. So this was the very first one. And it's because people started to try to isolate it directly. And there are a few cultures now, but up until quite recently, no cultures. So then the question is, are members of this family, the Muribaculaceae, are they friend or foe? So we see that they're often significantly associated with disease states, such as colitis, inflammatory bowel disease, and intestinal inflammation, also graft versus host disease, which is very nasty, and diabetes. And if you use IgA labeling, some family members get coded, which indicates that they are activating the immune system. And also, if you look in the genomes, they encode peptidases, fimbriae, type 9 secretion systems, which are usually associated with ways that bacteria evade our immune systems or activate our immune systems. However, as I mentioned before, they're mostly observed in mouse models. So the question was, how relevant to human disease are the Muribaculaceae, given that they have much lower overall incidence? So the way we began to investigate this was to use the evolution aspect, to try and get a look at how Muribaculaceae have evolved with mice and rodents more generally. So here is a timeline of rodent evolution. So it's estimated that the order Rodentia emerged somewhere around 90 million years ago, and then mouse and rat diverged around 10 million years ago. The common mouse that we use in the laboratory about 1 million years ago. And in fact, the laboratory strains that we use so commonly for research these days are only about 100 years old, and they're deliberately been inbred, which is not great generally, as we know from human genetics, to inbreed animals, but they're inbred so that their genetics are easy to interpret when we're trying to work out what's going on. So this domestication also involved, apart from host genetics, change in diet, a change in habitat, a change in social density. So the mice are usually kept at pretty high densities, and their behavior is also changed when they're in a mouse house. So all of these factors have the potential to influence the gut microbiome. So what we did, this is part 2 of the talk, we did a survey of the rodent gut microbiome. This was also work that was led by Kate. And what we were interested to know is the family Mirabaculaceae historically dominant in rodents? And more generally, what have the effects of laboratory domestication had on the rodent gut microbiome? So this is a tree of the rodent order Rodentia, and we see the mice and rats here within this family Muridae, but other members of this order include wood rats, voles, and capybaras, and even porcupines and guinea pigs and beavers. So it's quite a large lineage of organisms. So what we know from the literature, and this is a nice paper written in Cell by Rossart et al. In 2017, is that lab mice are really not natural. So this is a nice infographic for the paper. It's worth a read, that paper. But basically, what it's saying is that the immune system in a lab mouse typically has lower activation state than wild mice. And their immune system resembles babies, human babies. So it's not really prepared for its proper function in the wild. And then pet store mice resemble more, and wild mice resemble more the immune system of adult humans. There's a decreased diversity within lab mice compared to wild, and there was at this point some 16S studies that suggested that. And then they've also done studies where they co-housed pet store mice with lab mice, and that actually improved the innate and adaptive immune responses of the lab mice. So that's suggesting that some of this, the changes that occur within lab mice, can be compensated by microbiome being transferred from a pet store mouse to a lab mouse. So this is the study we did. We had 217 fecal samples, 50 from house mice, 126 from lab rats, that's 270 just from the lab mice, I might add. Then it was harder to get samples from these other groups, but we did it through collaboration and got some rodent samples from Lithuania, of all places. And so we had striped field mice, 16 of those, yellow-necked field mouse, 22 of those. So that's the Muridae family. For the voles, we got field vole, common vole, prairie vole, tundra vole, bank vole. Before we started this study, I didn't even realize there were so many voles, but there are a lot of voles. So we got a good sampling of those. Wood rats, we got 12 white-throated wood rat, Stevens wood rat, and desert wood rat. And these are amazing animals. They're incredibly tough. They can survive a lot. So they're a good one for comparison. And then everybody else, we got 3 beaver samples, 8 guinea pig samples, a couple of capybara samples, and 3 porcupine samples. So this was to give us a global view of the rodent order. So in total, we had 16 rodent hosts and 647 fecal and cecal samples. We used some of the there was public information as well that we tapped into. And then we did shotgun sequencing of these fecal samples, and then we obtained the genomes from that sequence data, and that's the so-called metagenome assembled genomes, or MAGs for short, which I'm sure many of you have heard about. So we got over 5,000 MAGs from this study. So when we then did comparative analyses of these different samples, and we just used the standard sort of principal component analysis type of studies. to see how different or dissimilar the microbiomes were. And so the way you read these maps is that each dot represents a sample, and then how close they are on the map tells you how closely related the microbiomes are to one another. And then you have principal component 1 and principal component 2. So these are the components that are accounting for the majority of the variance between the samples. And what we saw, as we have seen with many other animals, is that the host itself is a primary driver of the microbiome. So you see clustering of the microbiomes according to their rodent host. That is perhaps not so surprising. It's the same story between the fecal and the cecal samples, and we see that very commonly in gut microbiome studies when people compare different animals. This obviously, don't get too worried about this figure. It's pretty dense, but basically it's an overview of the bacterial diversity, so phylogenetic tree of the genome sequences that were found across all of the different hosts. So this is the central tree here, the different families. So there's Microbial here are highlighted with these different colors. So very common within the gut microbiome of animals are Lactospiraceae and Ocelospiraceae. So they are unsurprisingly also very well represented within rodents. And then all of these rings here, there are way too many to look at here, represent the different host types. So you can see that some areas are very prominent across all rodent hosts, and other ones tend to be specific to certain rodent hosts here. So, in summary, members of the bacterial families, Lactospiraceae and Ocelospiraceae, they're the most broadly distributed across rodent hosts. And there are some as yet uncultured genera that are particularly conspicuous. We don't have any isolates and we don't have any names, but we have these placeholder names, UBA 2821 and UBA 9475. We're present in almost all rodent hosts. So if I was interested for a project to find out about conspicuous species in general within rodents, these would be very good targets. Many bacterial species are widely distributed in rodents that are absent in humans and pigs. So I didn't mention here, but humans and pigs are here on the outside, the little purple rings. That's for comparison. And they're particularly conspicuously absent in Microbial is conspicuously absent in humans and pigs, and quite common within rodents in general. So we're starting to get an answer to that initial question. We could also estimate gain and loss of gut species that were associated with laboratory domestication. So this is, again, where you can put that evolutionary lens on the data. So we want to know when you had that massive change in the lifestyle of the lab rat, what happened to the gut microbiome? And so what happened, in short, that there were 452 species that were gained and 82 that were lost or below detection. Keeping in mind, of course, this is all about detection within these systems. Several families of clinical interest, including Streptococcaceae and Enterococcaceae, were inferred to have gained over half of their representative species in laboratory environment. Now, why might these ones be of clinical interest? Because often opportunistic pathogens belong to these groups. So it suggests that when mice were domesticated in the laboratory, there is associated with quite a large emergence of pathobionts. So let's take a closer look at the Microbial family again. So this is the relative abundance of the family, and then these are the different hosts. And so what we see is that it appears that this family is historically dominant in rodents. So it's not like it's just emerged recently in laboratory mice. So that's answered that first question. But the relative abundance is lower in wild mice compared to the rodent average. So in fact, it's more common across other rodent hosts than it is in wild mice. But then we see a higher relative abundance in laboratory mice, higher than in wild mice. So what's going on? So what's going on is that, hang on, I'll just cut to the chase here. Why is it that in laboratory mice you get very high amounts of Microbial and in the wild ones? And it's because what's happening is that you're getting blooms of particular Mirabaculaceae species. So what we're looking at here are different Mirabaculaceae species and their relative abundance in different populations of mice. So you see these large spikes that are occurring. What that's saying is that these are blooming within particular laboratory mice. So they're acting kind of like opportunistic pathogens. They're suddenly colonizing the mice within those laboratory facilities. So rather than having an even distribution of different species of the family, as you see in other rodents, you get this blooming effect within laboratory mice. So diversity lower because of Mirabaculaceae blooms in different laboratory cohorts. Laboratory-specific species carry genes suggestive of increased capacity for invasiveness or triggering of inflammation. So that means they're acting like pathobionts. And it's pointing again that the domestication of laboratory mice is associated with the emergence of pathobionts, and some of those pathobionts are in this family Mirabaculaceae. Okay, now I'm going to switch gears to marsupials, away from, we've heard enough about rodents, and I'm going to specifically talk about koalas. I'm sure everybody's familiar with koalas, apart from the fact that they're cute and they're cuddly, and that they have a very unusual diet in that they only eat eucalyptus leaves. And eucalyptus leaves, generally, you wouldn't think were a good choice of food source because they're filled with lots of nasty, toxic secondary metabolites, such as monoterpenes, sesquiterpenes, FPCs, cyanogenic glucosides, phenolics, the list goes on. So if we were eating eucalyptus leaves, we would get, in addition to not getting any nutrients out of it, we would get sick from it. And so the plant produces these toxic secondary metabolites specifically so that animals don't graze on them. But koalas and a few other marsupials have learned the trick of being able to digest the eucalyptus leaves, not only not making them sick, but actually being able to live on it. And they've got a couple of adaptations. One is the fact that they have a very long gut, so that the residence time is very long, so they have more time for the eucalyptus to be digested. And secondly, because you don't actually get that much energy out of eucalyptus leaves, they spend a lot of their time sleeping. So up to 60% of their time they're sleeping, and when they're not sleeping, they're resting. And so they only travel like 1% of the time. So they're very, very low maintenance animals. And also their brains are very smooth. So their brains are not using a lot of energy too. So these are all adaptations to this specialist diet. Okay, so we did a marsupial survey, and this is the third story I'm going to tell today. And Rochelle Sue was the postdoc that was, she's a group leader now, that was leading the study. And the question was, this is a tree of marsupials, and you can see you have koalas and wombats, which have a closest common ancestor here. And you have greater gliders, so koalas and greater gliders, they only eat eucalyptus leaves, they're specialists. But you have a few other things like ringtail possums and brushtail possums that eucalyptus is part of the diet, but not sole diet. And then you have everything like kangaroos and other macropods, they eat grass, right? So what it's saying is that this specialist diet has either arisen once in the past and then lost in other marsupials, which is very unlikely, or more likely it's evolved a couple of times within the marsupials that they've learned how to digest eucalyptus leaves independently. So we were interested to know about the adaptation to the eucalyptus diet, and it's occurred on multiple occasions, and is it independent microbial solutions, because we know the gut microbiome will play a role in digesting the eucalyptus leaves, and is that possibly due to horizontal transfer of eucalyptus adapted gut populations? So that was our question going into the analysis. Rochelle collected 92 fecal samples from 6 Australian marsupial families in the order Diprotodontia that comprise 23 distinct species. And the Diprotodontia has been around for estimated to be around 45 million years, so quite an, comparatively speaking, relatively recent. And the samples were collected from 4 wildlife sanctuaries and some from a campus kindy, in fact, here in Australia. And this is what the poo looks like. So koala poo is actually if I had to eat the feces of any animal, I would choose the koala because they smell good and they're like Tic Tacs. So they produce very nice fecal pellets. This is from kangaroos, just regular looking. And then the wombat's quite famous because it produces this cubic shaped poo, well known for it. Okay, so the methods, just in brief, sequencing of the fecal samples. And so we did the usual assembly of the metagenome assembled genomes and combine them with additional reference genomes from the GTDB. And so then we could do a taxonomic analysis of the data, but also a functional analysis, kind of as I already alluded to in the rodent study. But in addition, we did a metabolite analysis where we looked at the metabolite profiles. This is another overview tree showing the different genome sequences that were recovered from the marsupial survey. So it's colored by the phylum in this case. So again, we see a lot of members of the firmicutes, or as it's now called, the bacillota, which is very common in all guts. So marsupials are no exception. They also have these very common type of gut species. So we obtained about 2,000 mags that represent 1,758 species. And interestingly, even though the species novelty was quite high, so 81% of the species that we identified from the marsupials were novel, only 9% of the genera were novel, and in fact, only one family was novel. So this would suggest that despite the fact that marsupials have been isolated for a long time, there are very, very few gut microbiome studies. In fact, there is enough that we have looked at enough different gut microbiomes that we're starting to saturate the higher taxonomic level diversity of animal guts, at least what we can see in the high abundance population. We also found over 9,000 gene families and 360 carbohydrate active enzymes. And then the metabolites, we could detect over 300 metabolites that belong to 16 different classes. So as for the rodents and as for all the gut surveys so far, when you did principal component analysis, again, the host phylogeny is driving the diversity of the bacterial community too, the microbial community, which makes sense because they're kind of isolated within their host generally. So it's primarily driven by host phylogeny. So the colors indicate the different families, so kangaroos, wombats, gliders, and koalas. Secondarily driven by diet. And we see quite a bit of Mirabaculaceae in koalas, which is interesting, and particularly in captive koalas more than wild koalas. And then we see things like Ruminococcus, which is very prominent in things like greater gliders. If we look at the taxonomic composition by diet, so here we see the grass feeders. So we see a primary split between the marsupials that eat grass, like kangaroos and wombats, and then everything else, the grazers that eat like leaves and insects and other things are sort of on the right-hand side. If we look specifically at eucalyptus, we actually see the ones that are the primary source here, somewhat clustering together, but some separation as well. So if we look at the functional composition, again, the primary driver is host phylogeny, but the effect of the diet's more apparent. So now we see a greater clustering of grass feeders, and this may be due to functional convergence rather than taxonomic convergence. So different organisms, but the same kind of genes that you need, functional genes, to break down that food source. And that's pretty common where you have, as long as you have the function in your gut, in some ways it doesn't matter which organism is providing that function. So another PCA plot showing the functional composition by diet. So here again we see clustering of grass feeders, and then if we look at the eucalyptus, we see primary eucalyptus feeders like koalas clustering to some extent away from koalas and wombats, but also away from gliders. So the signal is still there from both the host phylogeny and the diet. So, and finally, I know there's a lot of PCA plots here. If we look at metabolite composition. So this is now the downstream side of the digestion of the food. We still see a host effect, so you still see some clustering, but it gets a bit murkier. We see metabolites from the eucalyptus leaves. I mentioned it's full of secondary, nasty, toxic secondary metabolites that drive primary feeders together, ones that are eating just eucalyptus leaves. And then we see some specific eucalyptus-derived metabolites that are identified. So in the koala, we saw things like eucalyptol, gamma-terpenine, and epiglobulol. And in the greater glider, which is the other main eucalyptus feeder, we saw things like menthol, alpha-cubabine, and alpha-limonene diepoxide. So what this is telling us is that the breakdown of the eucalyptus leaves is occurring separately within these two different hosts, and they're using different enzymes to produce different metabolites. But at the same time, there are many metabolites present in the eucalyptus leaves that would go straight through the animal, and that's what's driving together that's why their profiles look, based on the metabolite composition, they look similar to each other. Okay, so these are the metabolites that are common to eucalyptus. So these are the ones that are driving the profiles together. So the conclusion then, after all those PCA plots, is that eucalyptus feedings appears to have evolved independently in the marsupials using different gut species with some functional convergence. Was there a question?
Yes, Prof.
That's okay. I'll keep going. I just thought I saw a hand up for a minute. So this brings me to my fourth and last story for today's talk, and that is a planktomycete within the koala. This is again led by Rochelle. So I mentioned that there was only one novel family in the whole survey, and that was we were a bit surprised because planktomycetes are pretty unusual organisms, and they were found, some planktomycetes were found in koala poo. So this is a close-up of the phylum Planktomycetes, which is now called Planktomycetota. And the classical Planktomycetes, which if you've heard about them at all, these will be the ones you've heard about, are in the class Planktomycetia. And for instance, Gemata obscuriglobus, have people heard of that organism? My old PhD advisor spent his whole career looking at Planktomycetes like this. And they're of note because they have these unusual properties. They don't have peptidoglycan in their cell envelopes. They have this intracellular compartmentalization, including membrane-bound nucleoids. So they kind of look analogous to eukaryotic nucleus. They don't do binary division. They do budding cell division like yeast. They have endocytosis-like mechanisms where they engulf particles. That's probably helped by the fact that they don't have peptidoglycan. And so it's been proposed that the planktomycetes are somewhat of a missing link between prokaryotes and eukaryotes. That's what my old PhD supervisor, John First, that was his whole career, was trying to make that link. Now, what's happened is that the culture-independent sequencing, genome sequencing, has revealed that there's an awful lot more diversity within this phylum than we've known from isolates. So most of what we know is actually coming from this one class, a few cultures from this second class called Phycosphaera, and a few others from this class called Brocadia, which is well known because it does Anammox. So that's, if you've heard of this, that would be why you've heard it, Anammox. But there are all of these basal classes, including one called and that's why they don't have names, called SZUA567. And it was this SZUA567 that's in the koala gut. So we made some phylogenetic probes out of 16S so that we could visualize it using fluorescence in situ hybridization to have a look at them. And so this was images showing that they're rod-shaped bacteria. And we thought, well, we could look at this both in terms of what is it doing in the koala, but also what does it tell us about the evolution of the Planktomycetota. So first of all, we went back and made higher quality metagenome assembled genomes. So using short read sequencing, we had a mag that was belonging to this group was in 250 pieces, 4.3 gigabases. And after we combined long-read sequencing, nanopore specifically, I think, in this case, we could actually get genome back in a single contig, a single circular context. And of course, I'm sure I don't need to tell you guys that if the higher quality the genome, the more you can say about it. So this is a tree of the particular group, SZU567. So all of the ones that are found in koalas are within the family, SZU-A567. And then all of the ones related to this publicly available genomes are from environmental. So some interesting environments like deep sea sediments and other things like that. Within the koala itself, we estimate that there are 11 different species that are represented within the family. So quite a bit of diversity within the lineage. But all of those koala species belong to the single family of the same name. Then we can map against this tree different features of interest. So for the evolutionary context, we wanted to see, does it have the genes for peptidoglycan synthesis? Because you recall I just said that the classical planktomycetes like Gemata don't actually have peptidoglycan synthesis. So interestingly, these guys can produce peptidoglycan. They're missing a particular gene which is common to the whole phylum, A penicillin binding protein VAN-Y, VAN-XY. But we predict they should be able to produce peptidoglycan. Looking at the cell division and cell shape, you can look at genes like MRED, which is the shape determining gene, and they have that, which indicates that they should have rod-shaped morphology, which is consistent with the imaging that we did, where we saw quite nice large rod shape. And the other thing is that they have cell division genes. So I mentioned before that these classical planktomycetes in the crown, they do budding division, but these guys are more like regular bacteria that use binary fission. Electron transport chain, they only have complex 5, which is the ATPase for energy generation. So that suggests that they're fermenters and anaerobes. They do have superoxide dismutases. That's superoxide dismutase is a way of getting rid of oxygen. So if you're an anaerobe, you want something like this so that you can tolerate if oxygen comes into your environment. Superoxide dismutases are pretty uncommon within the koala gut microbiome. So it's like if you're living in the koala gut, it's anaerobic in there by and large, so you don't need to protect yourself from oxygen. It would suggest that these organisms are sometimes exposed to oxygen and can survive that. So in terms of what carbon sources they might use, we estimated that they could use trehalose, ribose, and xylose, which would be all carbon sources that would be fairly common within the koala gut. And ribose degradation is enriched, which is also unusual for other members of the koala gut microbiome. So at the moment, we're trying to isolate a member of this group, and we're using this carbon source as something that's a bit unusual to try and grow our target group and not grow everything else. They're predicted to be motile via flagella. We haven't actually seen that under the microscope yet. We're still looking for that. And they have particular aspects of that flagella that are common to the phylum, like they don't have the H and T rings, which are part of the flagella. But we predict that they have flagella genes. Associated with motility, chemotaxis is how the organism moves towards things using the flagella. So we predict that they've got chemotaxis genes that are moving them towards something. We don't know what they're moving towards, but it indicates that they're able to move around. And this is unusual in the gut. So there's a big selective pressure within the gut environment to throw away your flagella. One, because you don't really need to be motile because you're sitting within the gut. But secondly, because flagella are targets for the immune system. So they're a net deficit if you're an organism within the gut. This was an interesting one. So the honors student working on this got COVID, and so he put the samples into the fridge. And when he went back to look for these organisms, suddenly there were a lot more of them. So we thought, oh, that may indicate that these guys can grow at lower temperatures than the rest of the gut community. And in fact, we found cryoprotectant molecules, there are pathways to produce trehalose and glycine betaine, which can protect you from colder temperatures. And since this slide, we've also gone and checked using the genome, you can estimate the growth temperature, like with tools like Tome. And indeed, these guys have a temperature maximum that's a good 10 degrees Celsius lower than other members of that koala gut microbiome. So all of this is pointing towards that these guys must have come from somewhere else. And recent additions, or recent additions to the koala gut. So in summary, from the evolutionary context, the koala planktomycetes don't share the classical features, which is absence of peptidoglycan, budding division, and intracellular compartments, at least what we can tell from fish. And they, it appeared, our guess and our hypothesis is that these eukaryotic-like features must have arisen within the crown planktomycetes, within the most derived planktomycetes. From an ecological context, we predict that they're motile, oxygen-tolerant anaerobes that are cold-tolerant. So it suggests a comparatively recent acquisition to the koala gut microbiome. So then the question is, where did they come from? So when we looked, I showed you the genome tree, there were only the koala ones, and then things that are coming from deep sea vents, and so we thought, well, they're obviously, koalas aren't going to be getting them from deep sea vents. And also the other thing I didn't point out is that the wombats are their closest neighbor, and you do not see these guys within the wombats. So the koalas must have gained these since diverging from the wombats. And if you look in the 16S database, we saw a clue. And the clue is that you see koala sequences interleaved with cockroach and termite sequences. So that you find these guys in the hindguts of cockroaches and termites. So you say, well, how would a koala be getting this organism from a termite or a cockroach? Well, it turns out that koalas eat termite nests. This was a surprise to me. So you see this guy here happily munching on a termite nest. So they're getting something from the termite nest that they're not getting from the eucalyptus leaves. And apparently, this is a fairly common behavior amongst koalas. So our hypothesis is then that the planktomycete populations were obtained by eating termites. And only koalas do this. Wombats do not do this. Okay, so on that note, I will finish my lecture and just acknowledge this is the group that worked on the rodents, with Kate leading it, Sarah Knowles, Lema, Jeanette, and were international collaborators on the rodent survey, Antiope, Jeff, Nancy, Sean, and Phil, all looking at the immunological aspects of rodent guts. On the koala side, Rochelle is leading this together with Kate. Kate was also working on this. We have Danish collaborators. Oh, it's gone. Oh, gone. Okay. All right, that's it. Thank you very much for your attention.
Oh, that's really amazing, Prof. Very cute koala. I miss koala so much. Okay, so guys, welcome. If you have any question, yeah, we have some question here, and most welcome if you like to ask directly or just write it in the chat box. Okay, Afif Jati, student in Melbourne.
Yeah.
The time is yours.
You're
Thank
in
you so
Melbourne.
much. Yeah. So that was fantastic talk, Profil. So yeah, I also just want to say good morning to everyone there in Indonesia, especially to my former lecturer, I think, and they are there as well. So I was the alumni of the Faculty of Department of Agriculture Microbiology, and now I'm doing my PhD at Monash University. And yeah, I'm really interested in your talk, and it was so fantastic actually. And I have two questions for your talk. So first of all, maybe just a bit very basic and so very general. You mentioned some new, if I correct me if I'm wrong. So oh yeah, I also have been using your tools, GDDP, for several years ago, and I found
You
it was
appreciate that?
really, yeah. So I was found it really helpful. But I have some questions as well related to that. So you mentioned some new terminology about the taxonomy,
Yeah.
like something with the suffix ota, something like that. So
Yep.
would you mind sharing with us like briefly like what kind of update it was like or is it something that we have to concern now if we are working with metagenomic studies, something like that? And
Sure.
also, yeah.
So the first thing I'll say is that the
Mhm,
names, that's the
mhm,
nomenclature, right? So taxonomy is the
mhm,
structure. So we mainly concern ourselves with the taxonomy, the structure, the evolutionary structure. The names, there are groups, committees that are specialized in doing the nomenclature. So the one that looks after the bacterial and archaeal nomenclature is the International Committee for Systematics of Prokaryotes, so ICSP, and they look after Prokaryotic Code, or ICNP. So short is the acronym for that. And interestingly, and this surprised me as well, phylum has only recently been recognized as a formal rank in the prokaryotic code. So that's why the name has changed. So the way it works with the nomenclature, and this is across all nomenclatures, is that you have a uniform suffix for different ranks. So for instance, families have the suffix ACA, orders have the suffix ALES, and the proposed standard suffix for phyla is OTA, OTA, and that's where it's come from. I wrote, because I found this all very confusing, because we sort of stumbled into the nomenclature thing, which is very contested topic, I might add. Ecologists don't like nomenclature very much. I wrote a review a few years ago in IJS, in ISME journal, where I talk about that. So I refer you to that. I go into that in some detail, which is really for the benefit of microbial ecologists rather than nomenclature people, to explain why the names have been changing. What I will say though, because some people are going, oh, I hate the taxonomy and nomenclature because the names change all the time. What I would say is that the names will be stable now, because now we have these formalized naming conventions that are recognized in the nomenclature code. So we won't see a change of name for Bacteroidota anymore, right? So that's going to be the formal name that people will use moving forward. So what I'm doing and what I recommend to others is just to go with the new names, and they will stabilize.
So is there any consequence with established metagenomic tools that are already available outside there, or how could we like adapt this new system with those kind of tool?
So I've noticed that a lot of those tools are adapting to the new names and just using it.
Okay.
Yeah,
Okay.
I mean, mostly people don't concern themselves with the nomenclature. So
Mm.
tools that do certain things for metagenomics just use reference databases like the GTDB. So the GTDB
Mhm.
is moving to the new nomenclature, and so many other tools that use GTDB follow suit. And you'll see things like NCBI taxonomies move to the new nomenclature. So that's where everybody's
Mm.
more or less heading.
Okay. So and yeah, thank you so much for your answer. It's really enlightening. So my second question is now it's a bit technical in analysis. So outside there there are still pros and cons between like someone who only use like only 16S genes as metagenomic profiling, some say like that. And also like you have done like with metagenome assembled genomes. I was wondering in your opinion, if we want to capture or we want to get really good identification, let's say, or classification, is there any things that we should concern with those kind of thing like 16S, especially like 16S only sequencing or something like that, amplicon sequencing like that? Because I think we also have something like what may be very interesting to do in Indonesia, like in your case, like you did with koala. Maybe we have something like orangutans
Oh
in the
yeah.
wild. Yeah, orangutans in the wild life, as well as the orangutans maybe in the zoo, maybe something like that. So yeah, I was wondering if is there any like concern that we should know whether we before we start doing the sequencing, which one we need to know, like, or we have to be concerned, like if I use 16S only with specific primer, or maybe just go with the shotgun. So I just want to know your opinion about that.
So we use 16S as a first look because it's much cheaper than shotgun sequencing, right? Although the costs keep coming down all the time. It's easier to analyze as well, right? So we use 16S for a first look. And then after that, we may select a subset of samples to do the shotgun, the metagenomic sequencing on. The tools, unfortunately, the classification is still a little bit like there's a lot of differences between, say, silver or green genes, which is 16S-based data sets, and GTDB, but we're working with those resources to try to make it a consistent nomenclature and taxonomy. The things you have to be careful with the 16S, you've already put your finger on some of them. There's no perfect set of primers. So some organisms will always be missed by the primers. That just happens. So there's no way around that really. And the other thing is that the resolution, because particularly if you're using just a short 16S sequence, usually doesn't give you species level resolution. So for species level resolution, you really need to do the MAGs and the shotgun sequencing.
All right. Yeah. Thank you so much. It's very enlightening. Thank you.
No worries.
Okay, thank you, Prof. Phil. The next question, Gali, time is yours, please.
Okay, thank you, Ma'am Desi, for giving me the opportunity to ask my questions. Yeah, I have a few of these. So first of all, I would like to ask one of the first things that got me interested is that apparently the abundance of S24/7 is around 2% in humans, but it can be up to 70% in the hunter-gatherers. Now, I want to ask, first of all, like, what can influence the abundance of said S24/7 in us humans, you know? Also, what I also think is interesting is that the Muribaculaceae, I hope I pronounced it right, is often associated with...
That's right.
It's often associated with diseases in rats, but it can also, if I took note of it correctly, it can also interact with the immune system and possibly activating it. So I was wondering if there's a possibility of like genetic similarity between rodents and humans, so that possibly the same Muribaculaceae can have the same beneficial effects for humans.
Yeah, okay, so let's see. I'll go with your first question. I think it's very much because they have a lot of carbohydrate active enzymes. So I think modern, like urban humans, I should say, we don't get nearly as much fiber in our diet as our ancestors. I don't know about if that's the case in Indonesia, but certainly the case in Australia and places like the US, we have much less fiber in our diet. So as I showed you from the functional profiles, a lot of the Muribaculaceae species break down those fibers. So I think that's why you see them in higher amounts in more ancestral kind of living conditions, like with the Hasda people, because they eat a lot more fiber in their diet. And so I think that'd be my guess is the main reason for the higher amounts. And I think that's also the case with rodents, because rodents eat a lot of plant material, so they get a lot of fiber in the diet. This is one of their primary diets. So I think that explains overall a greater amounts of Muribaculaceae. I would imagine if we put an urban human on a very high fiber diet, that we would see increases in the Muribaculaceae within their gut microbiome. That would be an interesting experiment to do, for instance. As far as the negative effects of the Muribaculaceae, we certainly know that some species within the family, keeping in mind that that's a lot of species within this family. So you can't just say that every species is going to have an immune reaction, or every species is going to be breaking down a certain fiber, right? There's a lot of variation. But what I would say is that some subset of those Muribaculaceae species certainly interact with our immune system. They get coated with IgA, for instance. And I think that's what we're seeing in the case of the lab mice that have repressed immune systems, which allow organisms to flare up or grow up because their immune system's not keeping them in check. So I think that's a different aspect to the general effect of fiber degradation. You have some of those Muribaculaceae species which are capable of being pathobionts. In other words, organisms that can become pathogens under certain situations. And that's what I think is going on there. Does that answer your question? Okay.
Yes, that does answer my question, but I also have one more question. I hope you don't mind.
No, I don't mind.
Okay. So hearing the details about plantomycetes or plantomycetia, it's also interesting about the abnormality of it compared to the other koala cells. And looking at the results of the research between like a normal koala and the koala SZUA567, it's I think it's interesting how there's also a few reduction in the usually normal to find enzymes and other microbes. So I was wondering, also, since the koala also gains the plantomycetes from possibly eating the termite nest, is it possible that by eating the termite nest, they can also lose some of their normal enzymes and other nutrients, possibly? That's my last question, yeah.
It's possible. I mean, what I would say is that this is not the first time that we've seen particular bacteria get transferred from the gut of one organism to another. For instance, in a study we did of termites, we saw termites that eat cow dung. So there's particular termites that will live inside cow dung. They had some species that clearly came from the cow, from the rumen, from the cow microbiome. But what I would say is that this is unusual. I'd say most of the time, if you eat something, eat another animal's poo, which is very common in the animal world, by the way, coprophagy, most of those bacteria, because they're foreign to that gut, do not manage to survive in the new host. But a few, for whatever reason, do manage to survive, and then they can, and that's what's happened in the case of the koala. And not only have they, so we estimate, looking at the trees, that there's been at least 2 successful introgressions of Mirabaculacea. Oh, sorry, I'm getting confused now. The planktomycete bacteria into the koala gut, and that within the koala gut that then speciated, because we're talking about possibly millions of years here, right? Because the koala diverged from the wombat 15 million years ago, I think. So this could have happened, you know, just a few times or a couple of times successfully, anytime within that period. So does that answer your question? So yes, once you've managed, so keeping on with your question, once you've managed for a new foreign bacterial species to colonize a foreign host, then whatever capabilities that foreign microorganism carries, it can start to use in the new host. So that's how you pick up new functionalities. In fact, I think that was the case with the particular species that transferred from the cow to the termite that live in the dung, help the termite break down the dung fiber because they were more adapted to grass fibers. Does that answer your question?
Yes, it does. Thank you, Professor.
Thanks for the questions.
Thank you, Tejo. Tejo is our new student in undergraduate, Prof. So actually, we still have question. Do you still have time, Prof?
Yeah, I'm okay. If you want,
okay.
I'm happy to take more questions. If you guys,
Yeah.
I don't want to eat into your day too much, but I'm fine.
Oh yeah, okay, thank you. So we have question on the chat box here from Yeni Indra. She is a PhD student in our university. Thank you for your great presentation, Prof. I want to learn more about how flagella in the gut microbiome can be immunogenic. Can it also happen in plant microbiome, please?
That is a very good question to which I do not have a very good answer. We know, and there's probably a big literature on this, and so I won't be able to answer this very well, but we know for sure that you can actually visualize that the flagella are targeted by immune cells because they've taken, you can break off the flagella and look for the immune response, for instance, and you can also visualize it with fluorescently stained antibodies, so you can see that the flagella becomes a target. And presumably the immune system picks flagella because it's very different from things that it encounters from. So you don't want your immune system to target your own cells, and we don't have anything that are like flagella in our own bodies. That's my guess. I may be way out of line here, so I would definitely follow up and look in the literature on this. So it's something that can be targeted, because the other thing is that often pathogens have flagella, so that's a good target if you're trying to take out pathogens as well. The interesting question about whether plant immune systems may also recognize flagella, I have no idea at all. I think that's a really interesting question, and that's definitely worth looking in literature or asking ChatGPT if that's the case.
Yes,
I know
thank you
there
very
are
much.
similarities between plant and animal immune systems and differences, so it's quite possible that maybe it can target flagella as well.
Okay, Prof. Next question is from Nurfianto, from Malang, close to Surabaya anyway. Yeah, so the question is, I want to ask Prof. Phil, can you give us example or experience the effect of evolution and the dynamic of gut microbiomes between domesticated animals, including pets and livestock, and their wild relatives, to their health and also productivity? Thank you.
So I think the example I gave with the termites that live in the dung is one example how organisms have moved horizontally between a domesticated livestock, cows, into termites which are feeding on the dung. So that's one example. We know that cohabiting species like us with our pets, dogs and cats, and also the housing, there's a lot more shared microorganisms, not only from the gut, but also on the skin. So we know that these organisms can be shared. But again, it comes down to, is it transient? Are these organisms that are just moving? So if my dog licks my face and I get some of her gut microbiome, most of those organisms are just going to go straight through my system, right? But maybe some of them are able to survive in my gut, so therefore I have a transfer between me and a dog. Another, I guess, example, in general domestication, including our own domestication of our own species, humans, has resulted in reduction in diversity, particularly if what we're eating, like if we're raising animals that have just fed grain and not, say, grass, you'll see a reduction in their diversity. That's a result of the domestication. So did that answer that question, covering a lot of ground? There is, I'll also point out, there's a ton of literature out there on these type of questions. So I would refer the questioner to have a look as well. That's the first thing I can think off the top of my head.
Yep. Thank you so much. Is there any other question, guys, if you have? Yeah, if not, maybe my question will be in the last one. So, Prof, I just got to know that the koala is eating termite. That's so funny.
Yeah, that was a surprise to me too.
Oh, yeah. And based on your presentation, you mentioned that the composition of their life is 90% of sleeping and 20% of resting. Oh dear. Yeah. Do you think that that kind of behavior is also influenced by the gene or maybe by the microbes inside the gut?
Yeah, so this comes down to the question of cause and effect, which is always really difficult to work out what is the cause and what's the effect. I think the cause is that the food source is very poor and very toxic. So the effect then is that you get bacteria in your gut that are able to contribute to that in some way, whether it's detoxification, whether it's being able to break down carbon source to provide a little bit of energy. The reality is that the amount of energy is still pretty low. And so then the effect is on the animal, it changes its behavior. It goes much slower, it sleeps a lot, it doesn't have a very fancy brain. And these are all adaptations that are to the feed, to the eucalyptus leaves. So it's affected both its behavior, its physiology, and its gut microbiome. Yeah,
Oh, that's really nice. So when we are sleeping too much or resting too much, maybe we need to see our food then.
that's right. Maybe we're eating too much eucalyptus.
Yes, yeah, yeah, maybe. Okay, guys, do you still have question? If not, Prof, do you mind if we take picture together on Zoom, please?
Yeah, sure.
Yeah. Okay, guys, please turn on your camera and turn on and using the background. And Ibu Santi, can you please take picture? Let's take picture together here. And we really hope, Profil, that we can continue our collaboration between UQ and Gajah Mada in the
Yeah, me
future.
too.
Yeah.
Yeah.
Have you ever visited Yogyakarta
I
anyway?
have not, and
Yes, please.
I would like to do that sometime.
Yes, please visit our university. Next collaboration, we might invite you in the next collaboration then. Okay, Ibu Susanti, can you please direct the photo session, please?
Okay, thank you, Bu Desi. Hello, everyone, and hello, Professor Phil. Let me take a picture for you. We have 5 slides, so we will take a picture for the first slide. Can you give us the best smile? 1, 2, 3. Okay. And wait, please wait for a moment. And the second one. 1, 2, 3.
I'm from microbiology.
Yes.
Ah, nice.
Yes.
And the next one. 1, 2, 3. Smile. Okay, actually, there are so many students that didn't open the camera, but it's no problem. Okay, and the last one. Okay, give us your thumb.
Oh,
Okay, 1, 2, 3. Okay, nice. Thank you, everyone.
thank you so much, Dr. Susanti and Prof. Phil. Really, thank you so much for your time. I know that you are really busy, but you are still making the time for us here together. Not only for our student actually here, but I was watching that a lot of lecturer and also scientists joining this morning lecture. Thank you so much, Prof. Phil. Maybe you have a last motivation for us, Profil, because we know that your H index is 130. That's really
Yeah, that was a funny poster.
yes.
Yeah, please burn that. Don't show that to anybody. Um,
Okay.
well, first of all, it was my pleasure to give a talk, and secondly, thank you to everybody that gave me their time to listen to my talk. The only thing I would say is, if you're a student, I started, in Australia. In fact, next year I'll have been started my studies at this university 40 years ago, and as a young student, I felt like I was a long way away from where all of the big science was happening in Europe and in America. But if you have an interest, and there's always a way that you can conduct the research. You guys look to be at a very good research institution. So just follow your interest and your passion, and find good supervisors who share your interest, and you can make it and get an H index of 130.
Yeah. Yeah, that's right. We don't have anyone here with that high anyway.
Well, it'll come.
Yes.
Yeah.
Yeah, maybe some of us. I mean, yes. Thank you so much, Profil. I noticed that find your interest and passion, and then find good supervisor, and you'll be there in 130 X index.
There you go. Yeah.
Yes. Thank you so
All
much,
right.
from Profil, and I hope this is not the end of our collaboration. There are still more to go, especially in research. I watched that you are having Asia foundation as well, so we can collaborate through that funding in the future.
Yeah.
Prof.
Let me know. Send me details. So that'd be good.
Yeah.
Yeah. And I would like to visit at some point. That'd
Yes,
be good too.
please. Yes, please, Gajah Mada, Prof. Universitas Gajah Mada in Yogyakarta. Not far from Surabaya anyway.
No.
Yes. Okay. Thank you so much, Prof. Phil, and everyone here. And I hope you have a very good day and the remaining day. And thank you very much, Prof. Phil.
Thank you. See you all.
See you. Terima kasih Bu Santi dan semua mahasiswa serta Bapak Ibu yang sudah hadir.
Terima kasih Bu Desi.
Jangan jadi koala ya. 60% hidupnya untuk tidur. Oke, selamat melanjutkan impian, passion, dan harapan. Semoga bisa H indeksnya 130. Amin.
Kita
Terima
love
kasih
you
semua.
juga.
Ya, terima kasih. Ada Pak Doni juga tadi. Pak Doni
Terima kasih,
dan
Bu.
Terima kasih,
ya,
Bu.
terima
Terima
kasih
kasih.
banyak.
Terima
Iya,
kasih, Bu.
selamat beraktivitas. Bye.